Welcome to the deep dive. Today we're tackling a molecule that, well, it gets a lot of attention, often not the good kind, cholesterol.
That's right. Say cholesterol. And most people think health warnings, maybe food restrictions.
Exactly. But the reality is cholesterol is absolutely vital. It's a structural hero in our cells, and it's the starting block for so many crucial things our body makes. A real biochemical superhero, you could say.
Definitely. It's fundamental.
So our mission today is to, cut through the noise. We want to really pull back the curtain on cholesterol's journey. How do you get it? How does your body make it? How does it travel around? What happens to it?
And we'll explore its critical roles beyond just being the bad guy. We're talking hormones, vitamin D, all sorts.
Right. By the end of this, you'll hopefully have a much clearer picture, maybe even change how you think about this molecule. Let's get into it.
Okay, so beyond the headlines, what's cholesterol actually doing?
Well, structurally, it's key for cell membranes. Gives them stability, flexibility. Think of it like mortar between bricks.
Okay, so holding cells together. What else? It's also the precursor, the starting material for really vital compounds. Bile salts for digestion, and all the steroid hormones.
All of them.
Yeah.
Like cortisol, testosterone.
Yep. Cortisol, aldosterone, testosterone, estrogen. All start from cholesterol.
Yeah.
Plus, it gets converted into other important things like ubiquinone, dolicho, and even active vitamin D.
Wow. Okay, so it's busy. But you mentioned it hates water, right? It's insoluble. How does it get around in our blood, which is mostly water?
That's the big challenge. It can't just dissolve. So the body uses specialized carriers. Think of them as microscopic delivery trucks called lipoproteins.
Lipoproteins, okay.
And cholesterol rides in these trucks in two forms. There's free cholesterol, which hangs out on the outer shell, kind of near the water. And then there's cholesterol esters cholesterol with a fatty acid attached tucked away inside the core, hidden from the water.
Got it. Delivery trucks with special cargo inside and out. So where does this cholesterol cargo come from initially? The intestines, right?
Right. Your intestines are handling cholesterol from two main sources. About, say, 300 milligrams a day might come from your diet.
Okay.
But, and this surprises people, your liver actually pumps about 1,000 milligrams, a whole gram of cholesterol, into your bile, which also ends up in the intestine.
Whoa, so way more comes from bile than from food on average.
Often, yes. And from that combined pool, dietary, and biliary, your gut absorbs roughly 55% into your bloodstream.
Is that just passive? Does it all just soak in?
No, not at all. It's regulated. Your intestinal cells, the anerocytes, they have control mechanisms. There's a really important system to pump out excess cholesterol or unwanted things like plant sterols back into the gut.
Pump it out. How does that work?
It involves specific proteins, ATP-binding cassette proteins, or ABC proteins for short. Think of apoCII and apoCII as tiny molecular pumps actively pushing sterols out of the cell.
Like bouncers at the door of the intestinal cell, deciding who stays and who goes.
That's a great analogy. And you can see how vital these bouncers are if you look at a rare genetic condition called phytosterolemia or pseudosterolemia.
What happens there?
If those ABCT5 or G8 proteins are defective, the pumps don't work. Cholesterol and plant sterols build up inside the enterocyte and then spill out into the blood. Levels get sky high.
And that causes problems.
Oh, big problems. A much higher risk of cardiovascular disease. It really highlights how crucial that efflux pumping system is for protection.
Makes sense. And knowing about this absorption process, has that led to treatments?
It has. There's a drug called ezetimibe. It works by blocking cholesterol absorption by the enterocyte. It targets a different protein, NPC1L1, which seems to be the main gate for cholesterol trying to get in.
So block the indoor, not just rely on the out pump.
Clever. Right. Less dietary cholesterol gets absorbed into the body.
But wait, if you block absorption from food, wouldn't your body just make more of its own cholesterol to compensate?
That's exactly the feedback loop we see. Your body senses less coming in from the diet and thinks, uh-oh, potential shortage. So it tends to ramp up its own internal synthesis.
So ezetimibe alone might not be enough?
It often works better when combined with another type of drug, one that also blocks the body's internal cholesterol production. Hit it from both sides, absorption and synthesis.
Okay, let's talk about that internal synthesis then. How does the body actually make cholesterol? Where do the building blocks come from?
It all starts with a very common molecule, acetyl coenzyme A, or acetyl-CoA. Every single one of the 27 carbon atoms in cholesterol ultimately comes from acetyl-CoA.
And where does acetyl-CoA come from?
It's versatile. Your body can make it from breaking down glucose or fatty acids or even some amino acids.
So lots of sources. And the synthesis happens where?
Primarily in the cytoplasm of the cell. And it's a complex process, takes quite a bit of energy in the form of ATP and reducing power from NADPH. It happens in roughly 4 main stages.
You get stage 1.
Stage 1 is about making a key intermediate called mevalonate. 2 acetyl-CoAs join, then another one adds on, forming something called HMG-CoA hydroxymethylglutarol-CoA.
HMG-CoA, that sounds important.
It is, because the next step is the conversion of HMG-CoA to mevalonate. And the enzyme that does this, HMG-CoA reductase, is the star of the show.
Why is that?
Because this step is the rate-limiting step. It's the main control point, the bottleneck, for the entire cholesterol synthesis pathway. It's highly regulated.
The master switch. Got it. So after mevalonate is made.
Stage 2 involves converting mevalonate into activated 5-carbon units. Think of them as energized Lego bricks. These are called isoprenes. And interestingly, these aren't just for cholesterol. They're also used to make other vital things like coenzyme Q, important for energy production, and dolichol.
Multi-purpose building blocks. Okay, stage 3.
Stage 3 is assembly. These 5-carbon units link up head to tail, first forming a 10-carbon molecule, then a 15-carbon one called farnesyl pyrophosphate. Then 2 of these 15-carbon units join head-to-head to make a 30-carbon molecule called squalene.
Squalene. Wasn't that first found in sharks?
That's the one, first isolated from shark liver oil. And these intermediate pyrophosphates, like farnesyl pyrophosphate, also have other jobs, like helping anchor proteins to cell membranes.
Cool side jobs. Okay, final stage. Stage 4.
Stage 4 is cyclization. Squalene gets folded and converted into a rinse structure. The first one formed is called linosterol, which already has that characteristic 4-ring steroid nucleus. Then several more reaction steps modify linosterol into the final cholesterol molecule.
Wow. From simple acetyl-CoA all the way to complex cholesterol. Quite a journey. And you said HMG-CoA reductase, that enzyme at the start, is the key control point.
Absolutely. Controlling its activity is crucial for keeping cholesterol levels balanced. And
Wow.
clinically, this is the enzyme targeted by statin drugs.
Ah, okay. That's how statins work. So how does the body regulate this enzyme?
It uses multiple layers of control. One is transcriptional regulation, controlling how much of the enzyme is made in the first place. Special proteins called SREBPs, sterile regulatory element binding proteins. When cholesterol levels inside the cell are low, SREBPs travel to the nucleus and basically tell the cell's DNA to make more HMG-CoA reductase enzyme.
So low cholesterol signals make more enzyme. What if cholesterol is high?
If cholesterol levels are high, SREBP is kept locked up in the endoplasmic reticulum so it can't activate the gene. Less enzyme gets made.
Clever feedback. Any other controls?
Yes. High cholesterol levels can also make the existing HMG-CoA reductase enzyme itself more prone to being broken down. That's proteolytic degradation. So get rid of the enzyme faster.
Okay, so control production and control breakdown. Anything else?
There's also covalent modification. The enzyme can be switched on or off by adding or removing a phosphate group.
Phosphorylation.
Exactly. Things that signal low energy, like the hormone glucagon or low ATP levels, cause the enzyme to be phosphorylated, which turns it off. Cholesterol synthesis uses a lot of energy, so the body shuts it down if energy is scarce.
And high energy signals turn it on.
Right. Insulin, signaling abundant energy, or high ATP levels lead to dephosphorylation, which turns the enzyme on. Makes perfect sense, biochemically speaking.
It really does. So once cholesterol is made, mostly in the liver, you said.
Yes, liver is the main site, though the gut, adrenal cortex, and gonads also make some.
Right. So what does the liver do with the cholesterol it makes or receives?
Several things. Some is used right there in the liver for its own cell membranes. A lot gets esterified. That fatty acid gets attached by an enzyme called apoCII and stored or packaged into VLDL particles.
Those delivery trucks again, to send cholesterol out to the rest of the body.
Exactly. The liver can also secrete cholesterol directly into bile, just as free cholesterol, or it can convert the cholesterol into bile acids.
And you mentioned a feedback loop related to diet.
Yes. If your diet is low in cholesterol, the liver ramps up its own synthesis. If your diet is high in cholesterol, that signals the liver to suppress its internal synthesis. It's constantly trying to maintain balance.
Let's talk more about those bile acids, or bile salts. You said they're made from cholesterol in the liver.
Correct. It involves several steps, adding hydroxyl groups, shortening the side chain. The very first step is catalyzed by an enzyme called 7-alpha hydroxylase. This is the rate-limiting step for bile acid synthesis.
Another control point.
And it's regulated too. High concentrations of bile salts themselves actually inhibit this enzyme, another feedback loop.
Smart. So the liver makes these primary bile acids. What are they called?
The main ones are cholic acid and chenodeoxycholic acid.
Okay, but they aren't quite ready for prime time yet.
Not quite. To make them really effective detergents for digestion in the watery environment of the gut, the liver conjugates them, attaches either glycine or taurine. This forms bile salts, like glycolic acid or taurocolic acid.
And why does that help?
It significantly lowers their pK, meaning they become much more ionized, more water-soluble, and much better at forming micelles and emulsifying fats at the pH found in the intestine.
So they become super detergents. And their job is...
They get stored in the gallbladder, released when you eat fat. In the intestine, they break down large fat globules into tiny droplets, that's emulsification, and they form micelles, these tiny aggregates that help ferry digested fats and fat-soluble vitamins across the intestinal lining so they can be absorbed. Essential for fat digestion.
And then what happens to the bile salts? Do we just excrete them all?
No, the body is incredibly efficient. There's a system called the enterohepatic circulation. Over 95% of bile salts are reabsorbed, mainly in the final part of the small intestine, the ileum.
95%. That's amazing recycling.
It is. They travel back to the liver via the portal vein and get reused, maybe several times for a single meal. However, that small percentage, less than 5%, that isn't reabsorbed.
Yes.
That gets excreted in the feces. And since the body can't break down the basic steroid ring structure of cholesterol itself...
This is how we actually get rid of cholesterol.
Exactly. The excretion of bile salts is the primary route for eliminating excess cholesterol from the body. Some cholesterol is also excreted directly, but bile salt loss is the main way. Gut bacteria can also modify the bile salts that escape reabsorption, making them less soluble secondary bile salts.
Fascinating. Okay, let's circle back to those lipoprotein delivery trucks. We know they carry cholesterol and fats. Can you describe their structure a bit more?
Sure. Picture a spherical particle. The inside, the core, is hydrophobic. It hates water. That's where the cholesterol esters and triacylglycerols, main dietary fats, are packed.
Okay, the oily stuff inside.
Then there's an outer shell that's more water-friendly, more polar. This shell is made mainly of phospholipids and proteins called apolipoproteins, or APOS for short. And some free, unesterified cholesterol is embedded in this outer shell too, helping stabilize it.
And these apolipoproteins, they do more than just provide structure.
Oh yeah, they're crucial. They help make the whole particle water-soluble, obviously, but they also act as cofactors, activating enzymes involved in lipid metabolism in the blood, like apoCII activating lipoprotein lipase, LPL.
LPL, what does that do?
LPL is an enzyme on the surface of blood vessels, mainly in muscle and fat tissue. It breaks down the triacylglycerols inside lipoproteins, releasing fatty acids for the tissues to use or store.
Okay. And the APOs have other jobs.
Yes, they act as ligands, like keys that bind to specific receptors on cell surfaces, telling the cell, hey, here's a delivery of lipids for you. apoE and apoB100 are key examples for receptor binding.
Got it. So let's run through the main types of these lipoprotein trucks. You mentioned chylomicrons first.
Right. Chylomicrons are the largest and least dense lipoproteins. They're made in the intestine after you eat a fatty meal. Their main job is to transport dietary lipids, triglycerides, and cholesterol from the gut into the bloodstream.
And they have specific APOs.
They start with apoB48, which is unique to chylomicrons. Then they pick up apoCII and apoE from HDL particles circulating in the blood.
Why do they need those?
apoCII is the key to activate LPL, so the triacylglycerols can be unloaded to tissues. As they lose fat, they shrink and become chylomicron remnants. Then, apoE acts as the signal for the liver to recognize and take up these remnants via specific receptors.
Okay, so chylomicrons handle dietary fat. What about fats the liver makes?
That's where very low-density lipoprotein, or VLDL, comes in. VLDLs are made by the liver to transport newly synthesized triacylglycerols and cholesterol out to peripheral tissues. This is often called the endogenous pathway versus the exogenous pathway for chylomicrons.
And similar process. They get APOs activate LPL.
Pretty much. VLDL starts with apoB100, then gets apoCII and apoE from HDL. LPL digests its triacylglycerols. As it loses fat, it becomes a VLDL remnant, sometimes called IDL, intermediate density lipoprotein.
What happens to these remnants or IDLs?
About half of them are taken up directly by the liver using apoE receptors, similar to chylomicron remnants. The other half undergo further changes.
Changes like what?
They lose even more triacylglycerols, partly through the action of another enzyme called hepatic triglyceride lipase. They also transfer some components back to HDL. As they become denser and relatively richer in cholesterol esters, they transform into low-density lipoprotein, or LDL.
LDL. Okay, here we are, the bad cholesterol. Why does it get that reputation?
Well, LDL's main job is to deliver cholesterol to peripheral tissues and back to the liver. It does this by binding to LDL receptors using its apoB100 key. About 60% goes back to the liver, 40% to other tissues like the adrenal glands or gonads that need cholesterol. That part is normal and necessary.
So what's the bad part?
The problem arises when there's too much LDL circulating, maybe because production is too high or clearance is too low, or when LDL particles become modified, like oxidized.
Oxidized LDL.
Yes, if LDL hangs around too long or under certain conditions, it can get chemically modified. Now, your body has scavenger cells, macrophages, which are part of the immune system. These macrophages have receptors that recognize modified LDL.
And they take it up.
They do. But unlike the regulated LDL receptor, these scavenger receptors don't shut off when the cell is full of cholesterol. So the macrophages just keep engulfing modified LDL.
They just gorge themselves.
Exactly. They become packed with lipids, transforming into what we call foam cells. And these foam cells accumulating in the artery wall are a hallmark, really the starting point of atherosclerosis, the disease that clogs arteries.
Ah, okay. That clarifies the bad label.
Yeah.
Now, what about the good cholesterol, HDL?
High-density lipoprotein, HDL. It's synthesized mainly by the liver and intestine, and it also picks up components shed from chylomicrons and VLDL as they get broken down. It's dense because it's relatively rich in protein compared to lipid.
And its main job is?
Its claim to fame is reverse cholesterol transport. HDL acts like a scavenger, but in a good way. It goes around to peripheral cells, especially in artery walls, and picks up excess cholesterol that the cells don't need.
Like a cleanup crew.
Precisely. It uses a protein called ABCA1 on the cell surface to facilitate the cholesterol efflux, moving cholesterol out of the cell onto the HDL particle. Then, an enzyme within HDL called LSET esterifies that cholesterol, trapping it inside the HDL core.
So it grabs the cholesterol and locks it away inside itself.
Yes. And then the HDL particle, now loaded with cholesterol esters, can transport it back to the liver. The liver can then take up the HDL or transfer the cholesterol esters to other lipoproteins like VLDL or LDL remnants, which are then cleared by the liver. Ultimately, this allows the excess cholesterol to be excreted, often as bile salts.
That sounds incredibly protective against atherosclerosis.
It is. This reverse transport helps prevent the buildup of cholesterol in artery walls and reduces foam cell formation. That's why high HDL levels are generally considered protective against heart disease.
And are there conditions that highlight HDL's importance?
Absolutely. There are rare genetic disorders like familial HDL deficiency or Tangier disease caused by mutations in that ABCA1 protein.
The one that gets cholesterol out of cells.
Yes. Without functional ABCA1, HDL can't pick up cholesterol properly, it gets broken down very quickly, levels plummet, and patients suffer from very early and severe coronary artery disease. It really underscores the critical role of ABCA1 and HDL in protecting arteries.
Wow. And HDL interacts with other lipoproteins too, you mentioned.
It does. It's like a central hub. It donates apoCII and apoE to newly formed chylomicrons and VLDL. It also exchanges lipids with them, often trading its cholesterol esters for triacylglycerols, a process mediated by another protein called CETP, cholesterol ester transfer protein.
Does that exchange matter?
It can. For instance, if someone has high levels of triacylglycerol-rich lipoproteins like VLDL, HDL might end up taking on too many triacylglycerols and becoming less efficient at reverse cholesterol transport. This can lead to smaller, denser HDL particles, HDL3, which might be less protective than the larger, more buoyant HDL2 particles.
Complex interactions. Okay, so cells need to take up lipoproteins like LDL. You mentioned receptors. How does that physically happen?
It's a process called receptor-mediated endocytosis. The lipoprotein particle, say LDL, with its apoB100, binds specifically to LDL receptors clustered in specialized regions on the cell surface called coated pits. They're coated with a protein called clathrin.
Like designated docking base.
Exactly. Once bound, the pit invaginates, pinching off to form a vesicle inside the cell, carrying the receptor and the bound lipoprotein. This vesicle fuses with other intracellular compartments, eventually delivering the contents to lysosomes.
Lysosomes, the cell's recycling center.
Sort of, more like the digestion center. Enzymes in the lysosome break down the lipoprotein, releasing the cholesterol esters. Another enzyme then hydrolyzes the esters, freeing the cholesterol for the cell to use maybe for membranes or hormone synthesis. The LDL receptor itself typically gets recycled back to the cell surface to grab more LDL.
And you said the cell regulates how many LDL receptors it makes?
Yes, critically. The amount of free cholesterol inside the cell acts as a signal. If intracellular cholesterol levels rise, the cell responds by decreasing the synthesis of new LDL receptors. It puts fewer docks on its surface.
To limit further intake. Makes sense.
Conversely, if intracellular cholesterol is low, the cell does two things. It ramps up its own cholesterol synthesis via HMG-CoA reductase, and it increases the synthesis of LDL receptors to pull more cholesterol in from the blood.
And this is how statins have a dual effect. They block synthesis.
Right. Blocking HMG-CoA reductase lowers intracellular cholesterol. The cell senses the shortage and responds by putting out more LDL receptors. More receptors on the liver cells mean more LDL gets cleared from the bloodstream. It's quite elegant.
Let's apply this. You mentioned J with familial hypercholesterolemia, FH, high LDL. What's the root cause there?
FH is typically caused by a genetic defect in the LDL receptor gene itself. The receptors might be missing or don't function properly or don't make it to the cell surface. The result is that cells, especially liver cells, can't effectively clear LDL from the blood.
So LDL just builds up in the circulation.
Exactly, leading to very high LDL levels from a young age and drastically increased risk of premature atherosclerosis and heart attacks.
And the treatment for Anne? Statins.
Statins are a cornerstone. By inhibiting her cholesterol synthesis, they force her liver cells, even with fewer functional receptors, to try and maximize LDL uptake by expressing as many LDL receptors as they possibly can. And adding ezetimibe helps by reducing the amount of cholesterol coming in from her diet as well.
A two-pronged attack.
Okay.
Are there other receptors involved in lipoprotein clearance?
Yes. Another important one is the LDL receptor-related protein 1, or LRP1. It's structurally related to the LDL receptor, but recognizes a broader range of ligands, including apoE. So it's important for clearing chylomicron remnants and VLDL remnants IDL from the blood, especially after a meal.
Is LRP1 regulated by cholesterol like the LDL receptor?
Interestingly, no. Its synthesis isn't directly controlled by intracellular cholesterol levels. However, it is upregulated by insulin. So after a meal, when insulin is high, LRP1 levels increase, helping the liver clear those post-meal remnant particles efficiently.
And then there are those macrophage scavenger receptors we talked about.
Yeah.
The ones involved in foam cell formation.
Right. SRA1 and SRA2 are examples. They bind modified LDL, like oxidized LDL. And the key difference is, unlike the LDL receptor, their expression is not downregulated when the macrophage becomes loaded with cholesterol.
So the macrophage just keeps eating and eating.
Pretty much, leading to that foam cell formation, which is so central to atherosclerosis initiation.
Let's think about I&A. Type 2 diabetes, metabolic syndrome, high LDL. What's going on there? It sounds more complicated than just faulty receptors like in FH.
It often is. In poorly controlled type 2 diabetes, chronic high blood glucose leads to non-enzymatic glycation sugar molecules randomly attaching to proteins. This can happen to the apoB100 on LDL particles and to the LDL receptors themselves.
Glycated LDL and receptors, does that impair function?
Yes, glycated LDL might not bind as well to the receptor, and glycated receptors might not function optimally. This reduces the efficiency of normal LDL clearance. Plus, people with metabolic syndrome often have obesity and insulin resistance, which tends to increase the liver's production of VLDL in the first place.
So, more VLDL leads to more LDL, and the clearance mechanisms are impaired. A double whammy promoting high LDL and atherosclerosis risk.
Exactly. It's often a combination of factors in conditions like metabolic syndrome.
Okay, let's zoom in on atherosclerosis itself. Remind us of the artery structure.
Sure, an artery wall has three main layers. The innermost lining is the intima, mostly endothelial cells in direct contact with blood. The middle layer is the tunica media, containing smooth muscle cells providing strength and contractility. And the outermost layer is the adventitia, connective tissue.
And the first visible sign of trouble is the fatty streak.
Yes. That's essentially the accumulation of those lipid-filled foam cells, macrophages, in the subintimal space right under the endothelial layer. They often look like yellowish streaks on the artery surface.
What triggers this? Why do macrophages go there and start eating lipids?
It starts with injury or dysfunction of the endothelial cells lining the artery. Risk factors are key here. Things like high blood pressure, turbulent blood flow, high LDL cholesterol, especially modified LDL, low HDL, smoking, high blood sugar, inflammatory signals like angiotensin 7.
So these risk factors damage the lining.
They cause endothelial dysfunction. The injured endothelial cells then express adhesion molecules on their surface, like grab handles for circulating immune cells, particularly monocytes.
Monocytes.
Monocytes are recruited to the site of injury, squeeze through the endothelium into the subintimal space, and then transform into macrophages. These macrophages then start taking up lipids, becoming foam cells. The whole process is driven by inflammation. Atherosclerosis is now widely considered an inflammatory disease.
So it's not just passive fat deposition, it's an active inflammatory response gone wrong. What happens after the fatty streak forms?
The accumulating foam cells release more inflammatory signals and growth factors. This causes smooth muscle cells from the tunica media to migrate into the intima, proliferate, and start producing extracellular matrix components, like collagen. This forms a fibrous cap over the lipid core.
A plaque forms.
Right, a complex atherosclerotic plaque containing lipids, foam cells, smooth muscle cells, and fibrous tissue. This plaque grows, narrowing the artery lumen. But the real danger often comes later.
What's that?
The fibrous cap covering the plaque can become unstable, thin, and rupture. This exposes the highly thrombogenic core of the plaque to the bloodstream.
Thrombogenic, meaning it causes clots.
Exactly. Platelets rapidly aggregate at the rupture site, triggering the coagulation cascade, forming a thrombus or blood clot. If this clot is large enough, it can completely block the artery.
And if that happens in a coronary artery?
That's a heart attack, myocardial infarction. If it happens in an artery supplying the brain, it's a stroke.
So preventing plaque rupture is critical. Are there ways to stabilize plaques or prevent this?
Maintaining high HDL helps as it promotes cholesterol efflux out of the plaque. Lifestyle factors like exercise can raise HDL. Statins also have plaque stabilizing effects beyond just lowering LDL. And newer therapies are emerging.
Like what?
PCSK9 inhibitors are a good example. PCSK9 is a protein that normally causes LDL receptors to be degraded. By inhibiting PCSK9, these drugs allow more LDL receptors to remain on the liver cell surface, leading to very potent LDL lowering.
Fascinating. Okay, we've covered cholesterol transport and disease, but let's not forget its other vital roles, making steroid hormones and vitamin D.
Right, essential derivatives. Cholesterol is the sole precursor for all 5 classes of steroid hormones.
Remind us what they are.
Glucocorticoids, like cortisol for stress response and metabolism, mineralocorticoids like aldosterone for saltwater balance and blood pressure, androgens like testosterone, estrogens, and progestogens like progesterone.
Where are these made?
Mainly in the adrenal cortex, atop the kidneys, for cortisol, aldosterone, and some androgens. And in the gonads ovaries for estrogens and progesterone, tests for testosterone.
Is there a common first step?
Yes. Regardless of the final hormone, the very first step is converting cholesterol into a 21-carbon molecule called pregnenolone. This happens inside mitochondria and is catalyzed by an enzyme sometimes called desmolase, or P450-SCC. This is the rate-limiting step for all steroid hormone synthesis.
Another major control point.
Yeah.
Pregnenolone then becomes.
Pregnenolone is often converted to progesterone, another key intermediate. From progesterone, or pregnenolone itself, a whole series of enzymatic reactions, hydroxylations, oxidations, cleavage of carbon bonds, mostly by cytochrome P450 enzymes, leads down different pathways depending on which enzymes are present in that specific tissue.
So the set of enzymes determines the final product.
Precisely. The adrenal cortex zona fasciculata has the enzymes for cortisol. The zona glomerulosa has the unique enzyme needed for the final step of aldosterone synthesis, stimulated mainly by angiotensin II.
And problems can arise here too, like too much aldosterone.
Definitely. Primary aldosteronism, where the adrenals overproduce aldosterone due to a tumor or hyperplasia, causes the kidneys to retain too much sodium and water, leading to high blood pressure.
What about adrenal androgens?
The adrenal cortex, mainly the zona reticularis, also makes weaker androgens like DHEA and androstenedione. These can be converted to more potent androgens like testosterone in peripheral tissues.
Can things go wrong there? I remember the case of Vera L. with virilization.
Right. Vera had signs of masculinization. Her tests showed high levels of DHEAS and adrenal androgen. This often points to a problem in the adrenal gland. A common cause is a partial deficiency in one of the enzymes needed to make cortisol.
How does a cortisol deficiency lead to excess androgens?
If cortisol production is blocked, The pituitary gland senses the low cortisol and pumps out more ACTH hormone to try and stimulate the adrenals. But if the cortical pathway is blocked, all those precursor molecules get shunted sideways into the androgen production pathway instead.
Ah, so ACTH overstimulation pushes precursors down the open androgen paths.
Exactly. This leads to excess androgen production causing virilization. This is the basis of congenital adrenal hyperplasia, CAH, a group of inherited enzyme deficiencies in the cortisol pathway. Treatment often involves giving low doses of synthetic glucocorticoids to replace cortisol and suppress the excess ACTH drive.
Makes sense. And testosterone and estrogen synthesis in the gonads also start from cholesterol via pregnenolone.
Yes, stimulated by pituitary hormones LH and FSH. Leydig cells in the testes make testosterone. Ovarian cells make progesterone and estrogens using an enzyme called aromatase to convert androgens into estrogens.
Okay, last big derivative, vitamin D.
Also from cholesterol.
Sort of. It starts from a cholesterol precursor, 7-dehydrocholesterol, which is present in your skin.
So you need sunlight.
You do. UV light from the sun converts 7-dehydrocholesterol into vitamin D3, cholecalciferol. This is then transported to the liver, where it gets hydroxylated at position 25. Then it goes to the kidney.
Another step in the kidney. Yes, the crucial activating step. The kidney hydroxylates it again at position 1, forming 1025-dihydroxycholecalciferol, also known as calcitriol. This is the fully active hormonal form of vitamin D.
And this kidney step is regulated.
Highly regulated mainly by parathyroid hormone, PTH, and phosphate levels. PTH stimulates the kidney enzyme when calcium levels are low.
And what does active vitamin D calcitriol do?
It acts like a steroid hormone. It travels to target tissues, primarily the intestine, bone, and kidney, binds to nuclear receptors, and regulates the transcription of genes involved in calcium and phosphate transport and metabolism. Its main job is to increase calcium absorption from the diet and maintain calcium homeostasis.
So absolutely vital for calcium balance and bone health.
Critically, vitamin D deficiency, especially in growing children, leads to rickets, poor bone mineralization, skeletal deformities because they can't absorb enough calcium and phosphate.
Wow. Cholesterol really is the starting point for so much. Okay, let's try and pull this all together.
It's quite a story, isn't it?
It really is. Yeah. So key takeaways. Cholesterol is essential for those cell membranes. It's the precursor for all steroid hormones, for bile salts needed for digestion, and for vitamin D. It's definitely not just a bad guy.
Right. But because it's water insoluble, its transport through the blood is complex, relying on those lipoprotein particles, chylomicrons for dietary fat, VLDL for liver-made fat, LDL for cholesterol delivery, and HDL for reverse cholesterol transport, the cleanup crew.
And the synthesis of cholesterol itself is tightly regulated, primarily at that HMG-CoA reductase step, which is the target for statins. Absorption from the gut is also regulated via proteins like MPC1L1 and the ABC pumps.
Maintaining a balance, particularly keeping LDL levels from getting too high or modified and keeping HDL levels robust, is crucial for preventing atherosclerosis. That's really an inflammatory disease process triggered by endothelial injury and characterized by foam cell accumulation.
And we saw how genetic defects, like in the LDL receptor causing FH, or in enzymes causing CAH and virilization, or in the apoCII protein affecting HDL, can have really serious health consequences, driving the search for targeted therapies.
Exactly. Understanding these pathways allows for interventions like statins, ezetimibe, and potentially newer drugs like PCSK9 inhibitors.
Which brings us to a final thought. It's kind of amazing, isn't it? How a small change, maybe a tiny genetic variation in just one single enzyme like HMG-CoA reductase or one transporter protein like apoCII, within these huge interconnected biochemical networks, can ripple outwards and have such massive body-wide effects on your heart health, your hormone balance, your overall physiology. It really speaks to the elegance, but also maybe the fragility of our biochemistry.
A really profound interconnectedness. Well said.
Well, thank you so much for walking us through that complex world. And thank you for joining us on this deep dive into cholesterol. We hope you feel much more informed and maybe see this vital molecule in a new light.